Good morning to all of you. In the last class, we are discussing about the Hardy-Wimberg equation, a mathematical modeling which help us in understanding whether a population is evolving or not. To continue with this, the same thing today also, because all of you know that Hardy-Wimberg equation states that a population not uh, evolve if any population do not met these five conditions which you are observing now. Unevolving population. If there is no mutation in that one, there is no question of evolution. Even if there is no random mating, then also there is no uh, possibility of any evolution. If there is no natural selection, then also there is no evolution. And if the population size is extremely large population size, then also the chances of evolution will be very less. And if there is no gene flow between the two population, then also the population will evolve. So these are the conditions that to be met if you want a population to evolve. So then uh, one thing which we know that the Hardy-Wimberg equation also says that a population will not evolve unless it has been pressurized by some of the external factors. These external factors function as a agent or a tool to bring mutation or will force natural selection or random mating. So such things it will propose. So here a natural population can evolve in some loci while being in Hardy-Wimberg equilibrium at the other loci. What does it mean? See, evolution can happen at different loci on a gene. A Hardy-Wimberg equilibrium which will not allow the evolution to happen may be working on a particular loci. When it is working on a particular loci, a population can evolve at the other loci where the Hardy-Wimberg equilibrium is not operating. So that is a very interesting factor. Don't think that uh, if the Hardy-Wimberg equilibrium is functioning or it is with some loci, one loci of a gene, it will not allow that particular loci to evolve. Whereas the other loci, which is not influenced by this Hardy-Wimberg equilibrium, certainly can evolve. So now, applying this Hardy-Wimberg principle with one example, there is one locus which causes phenylketonuria. Phenylketonuria is a uh, genetical disease which is in the urogenital system. And in the now we can assume that the locus that causes phenylketonuria in Hardy-Wimberg equilibrium. Because here two conditions you know. The phenylketonuria PKU gene mutation rate is very low. Mate selection is random with respect to whether or not individual is carried with the phenylketonuria allele. See here it requires explanation. Phenylketonuria gene mutation rate is very low. Think that a particular person is having a phenylketonuria problem and this is at a particular locus. The phenylketonuria, the locus that causes phenylketonuria at a particular locus only on a gene. And the mutation rate of that PKU gene which causes phenylketonuria is very low. Now, person having phenylketonuria can undertake a random mating. When he undertakes the random mating, He may be mating with an individual who is not having the phenylketonuria. Also, there is a chance the individual is mating with the opposite sex who is carrying this PKU allele. That means either he will have a chance to mate with a perfectly normal individual or a person who is carrying this phenylketonuria gene. Now we will see what happens. The natural selection act can only act on rare homozygous individuals who do not follow any dietary restrictions. Okay. For phenylketonuria, there are certain dietary restrictions where one should not eat more protein and all those things are there. Natural selection can act on rare homozygous individuals who do not follow the dietary restrictions, who do not follow the dietary restriction meant for this phenylketonuria. Then equally the population size should be very large. Usually migration has no role in this phenylketonuria because many other populations also have the similar allele frequency. In any population you take, there will be some phenylketonuria patients. So that's why migration has not much effect or no effect you can say, as most of the populations have similar allele frequencies. Now what will happen? It has been estimated that this phenylketonuria is 1 per 10,000 birth. 1 per 10,000 birth. So Q equal to 0.01, Q square equal to 0.001. The frequency of normal allele is 0.99 because uh, 1 minus Q, 1 minus 0.1 equal to 0.99 because all of you know that the Hardy-Weinberg constant is 1. So P equal to 1, Q equal to, because 1 in 10,000 births. So that's why it is 0.01%. Q square equal to 0.001%. So P minus Q equal to 1 minus 0.01. So 99%, 0.99% I am talking the frequency of the normal allele. Now what is the frequency of carrier? That the 2PQ. 2 into 0.99 into 0.01, that is equal to 0.0198, or approximately in USA it has been observed that 2% of the US population is having this PKU allele. ಇದು have you followed this? ನನಗೆ ಏನು ಹೇಳಿದ್ದು ಅಂತ ಹೇಳಿ. Or if you have not followed anything, if you have not understood what I am telling, ಏನಾಗ್ತಾ ಇದೆ ಹೇಳ್ರಿ. ಮಧು, ಸೂರ್ಯಲಕ್ಷ್ಮಿ, ಹಲೋ, ಕೇಳ್ತಾ ಇದೆಯಾ? ಹಲೋ, ಏನು ರಿಪ್ಲೈ ಮಾಡ್ರಿ. ಏನಾಗ್ತಾ ಇದೆ? ಕೇಳ್ತಾ ಇದೆಯಾ? ಅರ್ಥ ಆಗ್ತಾ ಇದೆಯಾ ಇಲ್ವಾ? ಶ್ವೇತಾ ನೀವ್ ಹೇಳಿ, ಗೊತ್ತಾಗ್ತಾ ಇದೆಯಾ? ಏನ್ ಎಲ್ಲ ಅಥವಾ ಎಲ್ಲ ಮೊಬೈಲ್ ಆನ್ ಇಟ್ಟು ಎಲ್ಲ ಹೋಗಿದ್ದೀರಾ? ಯಾರು ಮಾತಾಡ್ತಾ ಇಲ್ಲ.
ಇಲ್ಲ ಸರ್ ಇದೆ.
ಈಗ ಏನ್ ನನಗೆ ಹೇಳಿದ ಅರ್ಥ ಆಯ್ತಾ ಅಥವಾ ಮತ್ತೊಂದ್ಸರಿ ರಿಪೀಟ್ ಮಾಡ್ಬೇಕಾ? ಹೇಳಿ. ಇಫ್ ಯು ಹ್ಯಾವ್ ಅಂಡರ್ಸ್ಟುಡ್ ಇಟ್ಸ್ ಓಕೆ.
ಮತ್ತೊಂದ್ಸತಿ ರಿಪೀಟ್ ಮಾಡಿ.
ಓಕೆ. So that's why I'm asking you. So this phenylketonuria occurs usually 1 per 10,000 birth. If 10,000 childbirth takes place, out of that 1 will be suffering from this phenylketonuria. So this is Q equal to 0.01. The frequency of normal allele. What is the frequency of normal allele? P equal to 1 minus Q. 1 is the, all of you know that it is the Hardy-Wimber constant. 1. If you add the all heterozygosity, it comes to 1. 1 minus 0.01 means 0.99 is the P value. That's the normal allele. What is the Q value, which is the allele for PKU? It is 0.01. Now, what is the frequency of carrier? That is 2PQ. That is P square plus uh, 2PQ plus Q square. All of you know that. Now, the frequency of carrier is 2PQ. 2 into 0.99 into 0.01. That comes to 0.0198. Approximately that comes to 2% of the population. In US it has been calculated, it is 0.2%. In India we don't have the exact data, but it says that it is around 1.99% as far as the recent study states. Okay, it is approximately 2%, we can keep it. Now, within this, what does it mean? The 2% of the US population, they are having this phenylketonuria, whereas 98% of the population is not having phenylketonuria. Now, going back to what we have discussed in the previous class, in spite of all these things, in spite of all these things, the population will carry this phenylketonuria gene to the into the future generation. Most of the time we think only 2%, only the dominance will prevail, and uh, oh sorry, so now any any population can get its frequencies of the alleles altered because of three things. One is the natural selection, genetic drift, gene flow. Three major factors, I should say, that can alter the allele frequencies and bring the most required evolutionary changes. What are these three things? One is natural selection, second one being the genetic drift, and the third one is the gene flow. Now, what is genetic drift? Natural selection I am not going to explain now again. In the end again we have to discuss about that. So a genetic drift describes how allele frequencies fluctuate unpredictably from one generation to the next generation. The frequency of the alleles, they generally fluctuate from one generation to other, and this fluctuation is unpredictable. Naturally, the smaller a sample, the greater the chance of deviation from the predicted result. If the population size is very large, then to a certain extent you can predict how the frequencies fluctuate. But if the population size or the population sample size is very small, means usually there's a greater chance of failing in predicting the result. We will not be able to see how the deviation from the predicted result appears. So, uh, a genetic drift generally tends to reduce genetic variation through loss of alleles. The main purpose, the main motto of the genetic drift is to reduce the genetic variation. That means how it will be able to reduce the genetic variation? By losing many alleles. More the alleles, variation will be higher. Less alleles means less variation. So genetic drift operates in such a fashion to reduce the genetic variation by losing many of the alleles. So here, see here, uh, the generation one, the frequency which we have already discussed, it is the dominant homozygous dominant is 0.7 and Q is the frequency of CW, that is the white flower, it is 0.3. In generation 2, it will be P and Q, 50%, 50%. But in the generation 3, all will be red and no. Even such things can happen by reducing the alleles. It is the result of genetic drift. Otherwise, what you will predict is if it is 7-3 in any generation, as per the Hardy-Weinberg equation that we have discussed, that will remain same irrespective of the dominant alleles and the size of the dominant allele and the recessive allele. But here it is not happening. When the in a small population, when the because we have selected only limited number of plants, that means the sample size or the population size is very very small. That is how the genetic drift operates. If it's a large population, of course, this is not going to be changed, it will continue. But when you have taken only a small population of few individuals, we will, whatever the result that we get is certainly most unpredictable. What we predicted will not hold good at all. Because it is because of the genetic. Then there is also one more possibility, that is the founder effect. What is this founder effect? Suppose from a big population, when few individuals gets isolated, it's a large population, but few individuals either they migrate to a new land or within the population they get isolated. Now, if you observe this isolated population, the allele frequencies of this small founder population, we call it as founder population because they have cut all relation from the main population and got isolated. Say they isolated and lands up in some island. Naturally, they become the founder population. And if you observe the allele frequencies in the small founder population, it is certainly different from those in the larger parent population. There's a lot of difference in the allele frequencies of the smaller founder foundation population and the larger parent population. This will cause bottleneck effect. What is bottleneck effect? The bottleneck effect is a sudden reduction in the population size due to a changed environment. If there's a sudden reduction in the population size due to a changed environment, the resulting gene pool may no longer be reflective of the original population gene pool. Same thing has happened in the founder population. There is a sudden reduction in the population size due to changed condition. They might have isolated, or somehow they landed in an island which they cannot go back to the mainland. And if you see pool, the gene pool is no longer the reflection of the original population gene pool. Because original population gene pool is much large. But whereas this isolated founder population gene is two this thing. This is what you call bottleneck effect because of the sudden reduction in the population size. And all of you know that if the population remains small, in a small population, the effect of the genetic drift is more visible and more pronounced. As a result of bottleneck effect, we got a founder population. And in the founder population being small, it is further affected by the genetic drift. See, this is the original population. This is the bottleneck effect. From large population, if you now the surviving population is this only this. So we can say that this gene pool is no longer the reflection of this gene pool. There's a lot of difference between the gene pool of this original population and the surviving population. And this surviving population is having very few individuals. As a result, the influence of the genetic drift on this is much high. Understanding this bottleneck effect, uh, we can increase understanding how human activity affects the other species also, because many times what we do is from a large population we isolate some other a small group of animals and we domesticate, or they are shifted to some other places. Everywhere this bottleneck effect. How? With the help of one example we can see that in a greater pyrie chicken impact of genetic drift, because as a result of a bottleneck population or bottleneck effect, population size of the founder has become very small. Naturally, if it happened, it has happened. What has happened? The loss of pyrie habitat. Pyrie is a grassland, which all of you know. There is a severe reduction in the population of greater pyrie chickens in one state of US, that is Illinois, because most of them died because of the severe reduction in the habitat, loss of habitat. Because pyrie habitat was the habitat for this uh, greater prairie chickens. The surviving bird had low level of genetic variation. It's also again bottleneck from a large population, a small population of greater prairie chickens survived. Most of the population has become extinct. Naturally, this surviving bird has low level of genetic variation. And as a result of that, what has happened is only 50% of the eggs used to be hatched. See, pre-bottleneck, this was the in the Illinois, the range of greater prairie chicken, which is shown in the green line. After the loss of prairie grass, now the range of greater prairie chicken is restricted to very, very small area. Naturally, here there were millions and millions of these prairie chicken. Now only few could survive. Just see here in Illinois, 1930 to 1960, the population was 1,000 to 25,000. In 1993, it was on less than 50. And number of alleles, when the large population was there, it was 5.2. And when the population size reduced, it is only 3.7. Percentage hatching, earlier it was 93%, but with the reduced population, it is around 50, slightly greater. And same is seen where in the canas, where there is no bottleneck effect, the population is about 7,50,000. Number of alleles is very high, 5.8, and percentage of a egg hatch is 99%. In another area in 1998, that is the Nebraska, the no bottleneck effect. Population size is maybe around up to 2 lakhs. Number of alleles are also around 5.8. The percentage of hatching is 96%. Now, what we are observed when the population size decrease, the number of alleles, the variation has also decreased, and this resulted in the percentage hatchability of the eggs. Now, let's move forward. So, great pile chicken. Researchers use the DNA from the museum specimen to compare the genetic variation in the population before and after the bottling. And they have observed that there's a loss of alleles at several loci. Researchers also introduced some prairie chickens from populations in the other states and were successful in introducing the new allele, and the egg hatching rate has increased to 90%. Now, what is the outcome? We are talking about the genetic drift. Genetic drift will not allow more and more variation. In a small population, the influence of the genetic drift is much higher. So that's why in this prairie chicken also, where the population size was minimum, genetic drift has become a very significant in small population. Naturally, the genetic drift makes all alleles, cause allele frequencies to change at random. This has resulted in the loss of genetic variation within the population. Most of the time it is observed the genetic drift can cause harmful allele to become fixed. As a result, in the genetic, here also the same genetic drift played its role. As a result, hatchability of the eggs came to only 50%. This is how the genetic drift brings down the variation and minimizes the possibility of evolution. Because all of us know now, increased variation means there's a lot of scope for natural selection. Now, already the genetic drift has brought the genetic variation to the lowest possible level, and it has fixed many harmful alleles also, and thus it has not allowed the population to show much variation because of the loss of alleles frequencies in the gene. So this is what is the effect. It is the effect of genetic drift. Now coming to the gene flow. Gene flow consists of the movement of alleles among the population, where there's a movement of alleles from one population to another population. What do you mean by that? Take the example of elephants in Karnataka. In Karnataka, we have two populations of elephants. One is in the southern Karnataka, that is Nagarhole, Bandipur, even up to Shimoga. And another one is another population of elephants in the Dandeli. Now, it is almost sure that these two populations are isolated. And there is no scope for flow of gene from Dandeli population to Nagarhole population because there is no elephant corridors. Suppose if these two elephants can mix means there can be a gene flow. But now the gene flow has been stopped. Gene flow is nothing but the movement of the alleles among the populations. And alleles can be transferred from one population to other population either by the movement of the fertile individuals or at least their gametes. Now, both the things are not possible because these are the two populations isolated. The gene flow also tend to reduce variation among the population over time. Gene flow will ensure the uniform distribution of the alleles in both the population, but and thus reduce the variation among the two population. If there is a gene flow between this Dandeli elephant population, Nagarhole elephant population, there is no scope for variation. Both will be same. But when there is no gene flow, there is a scope for both the population to evolve independently. There is a scope to evolve independently. So if the gene flow is there, then it becomes a larger population. And if they have to evolve means they have to evolve as a whole, considering all the individuals of the population. But a population, if it is isolated and there is still there's a gene flow, that means the variation will reduce. It is said that gene flow can decrease the fitness of a population. Because you know that survival of the fittest, the nature selects the best. Probably it will be hard to believe how the gene flow can decrease the fitness of the population and also to understand how the gene flow tend to reduce the variation among the population over time, not immediately over time. For this, we have to take an example of the great tit. This great tit is from a Dutch island of Leyland. How the gene flow takes place? When there is a random mating. Mating causes the gene flow between the central and eastern population in the population. There is a gene flow between the two populations of the same island. The immigration from the mainland introduces allele that decreases fitness. Why it's so? See now, both the central and eastern populations, the birds which have colonized in this island over a long period of time, coming from the mainland. And within the island, they got divided into two populations. One is central and eastern population. Whenever there's a chance for mating between the central and eastern population, the gene flow takes place between these two populations. But suppose if the immigrants come from the mainland, all of you know that mainland has more number of alleles, and that come and settles here in the island mean. Mainland has introduced, it has brought along with that many more alleles. But these alleles are not fittest alleles for an island life. Certainly, they are the fittest alleles for mainland life, but not for an island life. In this island, it has been observed that the central region has more high immigration, whereas the eastern region has less immigrants. It has also been observed that central region which experiences high immigration have lower fitness, whereas the birds in the eastern region with less number of immigrants, they have higher fitness. See, these are the survival rate. These are the surviving females, because uh, females are more important for us because they are going to participate in the breeding activity. Females born in the central population, survival rate is about 26%. But whereas in the eastern, it is survival rate percentage is around 23% in the central population. But whereas in the eastern population, see, the survival rate is even more than 50. So this is the central population and this is the eastern population. This is the bird. But we cannot every time say that gene flow reduces the fitness of a population. Even sometimes gene flow can increase the fitness of population also. Again, you may say that I am contradicting what I have told in the previous slide. What I want general, if you generalize it, gene flow will decrease the fitness of the population. But it all depends upon from where you get the genes. But sometimes the gene flow can increase the fitness of population also. Here you consider an example, the spread of allele for resistance to insecticide. The resistance shown to a particular any insecticide depends on the genetic makeup of the individual. Insecticides, especially the DDT, have been used extensively to kill mosquitoes which carry Nile, West Nile virus and malaria. It has also been observed alleles have evolved. In some population, that confer insecticide resistance to these mosquitoes. Some locus alleles have evolved on the locus, which start showing resistance to these insecticides. In some population, not in all population. Suppose if the flow of this insecticide resistance allele flows to other population, which will increase the fitness in that also. To make it more simpler, say there are two, three populations of mosquito. One is population A, population B, and population C. Mosquitoes were targeted, and so an insecticide was sprayed. When the insecticide was sprayed, an allele was evolved in population B, which confer resistance to this pesticide, so that the mosquitoes could survive even after eating the pesticide. The newly evolved allele is the most fittest character when the insecticides are sprayed. Suppose if this mosquito is able to interbreed, the one which is having better genetic makeup now, interbreed with the population of A and C, this resistant allele from the group B, population B, will flow to population A and C also, and make all the populations resistant to the effect of insecticide. It causes, that means here the flow of gene flow has increased, the gene flow has increased the fitness of the population. If the gene flow was not there from B, then eventually population A and C may die because of the repeated spray of the pesticide. But now population A and B also can survive because of the gene flow from population B, which has developed a pesticide resistance allele. So thus here the gene flow has increased the fitness of the population. As far as the human population is concerned, gene flow is an important agent in the evolution of human being or human population. Most of the human evolution is attributed to gene flow only. Now, after studying gene flow and genetic drift, we should again roll the role of natural selection over these two. And how the natural selection helps in the evolution. Evolution by natural selection involves both chance and sorting. It happens as a result of both chance and sorting. One is mere chance or selection. All of us know that new genetic variation arises by a chance. And we also know that beneficial alleles are sieved or sorted, and that has been favored by the natural selection. And ultimately, it is also true that only natural selection consistently results in adaptive evolution. Whatever the evolution is for adaptation. So, this adaptive evolution is the result of natural selection. How? The natural selection brings adaptive evolution by acting on the morphological makeup of the organism, or in other words, acting on the organism's phenotype. And all of us know that genotype is required for phenotypic expression, undoubtedly. Again, go back to what Darwin and Wallace have suggested. Struggle for existence and survival of the fittest. These are two misleading terms because they imply direct competition among the individuals. We think that it's a direct competition between the individuals. Struggle for existence and survival of the fittest. Many times there is no competition at all. When the insecticide was sprayed on a population of mosquitoes, here, when I say the survival of the fittest, there is no competition. Not because of the survival of the fittest, because of the competition, but it is because of its own genetic makeup with the invent of the new allele, which is providing the resistance. So that's why in the synthetic theory of evolution, we do not agree to the phrase like struggle for existence and survival of the fittest. More rationally, we use the reproductive success. And that's the main factor. And all of us know now that the reproductive success is generally more subtle and it depends on very many factors. Undoubtedly, the reproductive success is the most subtle one at the subtle level, at the very basic level. And it also depends on number of various factors. Here, it is not the survival of the fittest. What makes important is the relative fitness. The relative fitness actually contributes, it is the contribution by an individual where it carries the gene pool for the next generation. That means some individuals, by virtue of having or capacity of to produce more number of offsprings, they will certainly carry more alleles to the next generation. Whereas the one which produce less number of offsprings will carry less of these alleles to the next generation. The selection, the natural selection, favors certain genotype. How? Natural selection will not select the genotype. Basically, it is on the phenotype. By acting on the phenotype of certain organism. The phenotypic expression of those organisms is because of the specific genotype that they have developed. So here, natural selection favors or selects certain genotype, not directly, indirectly by selecting the phenotype, corresponding phenotype. Uh, Here it needs little more explanation. Probably now I'm running out of time. In the next class, I will be more. I will try to explain it little more and then go to the different types of selections.